much for the invitation to this really exciting event. I'm very glad to be here. So I'm going to talk about generative AI for antimicrobial peptide discovery. Is there a weird sound in the back or no? No, okay, sorry, it's just me hearing it. Okay, great. So first of all, why is this AI for antimicrobial resistance actually important? So unfortunately, for the last 30 years or so, there were no new classes of antibiotics discovered. And at the same time, resistance of bacteria to existing antibiotics is rising. And it was projected that by the year 2050, globally, antimicrobial resistance will exceed cancer and other major diseases as the main cause of death. So because of this, WHO and multiple research studies consider antimicrobial resistance a growing global health hazard. And now there's a lot of research going on around this topic, and one of the research lines that is being considered a promising solution is the design of antimicrobial peptides, shortly AMPs. And now for me, as a computer scientist, a peptide is actually a sequence of amino acids, so a string of letters like this. But actually what makes them work are their physical chemical properties, which are such that they allow these peptides, for example, to perforate the membrane of bacteria and kill them by causing lysis. On top of this, these AMPs are amazing for us because they are they target specifically negatively charged bacterial membranes, and they tend to be non-toxic to human cells, such as erythrocytes. So they are very promising, but they also pose a major challenge for designing new such peptides and searching the space of possible AMPs. And this is because sequences are combinatorial species. They are very hard to optimize, and it's very hard to traverse the huge space of possible peptides. So fortunately, many other so far unsolved challenges were recently cracked using AI. And we know about amazing advances in generating realistic photographs, text, and in the biomedical domain, predicting protein structures. So we reasoned that it could be that AI can be leveraged to fight antimicrobial resistance. And one way of doing so would be to use these new amazing generative models to design novel antimicrobial peptides. Now, the way these generative models work on a very high level is that they can be trained on very complex data, and they create convenient latent representations of the data, which are Euclidean vectors now. They are no longer, for example, like sequences, they are no longer combinatorial. And it's much easier to proceed, to traverse in such a latent space, which is Euclidean. Now, after training, these models can be used for generating very realistic artificial samples, for example, of peptide sequences. So now I'm going to show you our approach for doing this task. So a recently published generative AI model called HYDRAMP, which is an extended conditional variational autoencoder. So before going into the details of the model, let me tell you about the data that we used for training. So the data, first of all, compared to other large-scale data that is used for training such generative AI models, is very sparse. It's very scarce. So we had access to around 225,000 sequences of peptides. Okay, so we knew they have the property of encoding a valid peptide, but we didn't have any label for them. the labeled data set was relatively much smaller. So we had a bit over 11,000. Sorry, this just doesn't work. Okay, so you see this? We had 11,000 positive peptides and 11,000 negative peptides, meaning like we knew they are AMP or not. And then we also had even a smaller number of peptides for which we had their activity measured against target bacteria that we chose to work with. So E. coli, the activity is measured using minimal inhibitory concentration. And we didn't use that whole data set for training. We left out a test data set where we considered a bit over 1,000 positive, so AMP and highly active peptides, and a bit over 1,000 negative peptides. So not AMP, not highly active. All right. Sorry for this little funny shape. I actually corrected it at some point, but okay. So, okay. HiDRUMP has, now we're going to talk about how HiDRUMP works first on general level. So it has two ways of action. The first one is called unconstrained generation, and this is actually a very standard way of using variational autoencoders. So when you have the HiDRUMP model trained, it comes with a switch. So this red stuff is supposed to visualize a switch. The image got corrupted a little bit, but it is like a switch that allows you to tell the model, okay, please generate now a negative peptide, so not active, or generate a positive peptide. And the model is supposed to now produce these novel peptide sequences that satisfy the given conditions. This is how conditional variational autoencoders work. But now we also propose this analog generation mode, where apart from the model itself, you also introduce an input prototype peptide. So you give the prototype as input, you again switch that negative positive condition, and the model is generating novel peptides that are now supposed to satisfy the condition, but also be analogs of the prototype. So they're supposed to be similar to the original peptide. And for the input prototypes, you can consider, interestingly, not only positives, because you could imagine you're taking your prototype, which is a known AMP, and you want to use a model to improve it, right, to become more active, for example. But using negative peptide created for us amazing opportunities of actually generating very novel peptides, because they are similar to something that is known not to be active, and still actually after the model modifies it, becomes active. And so this increases the pool of the AMPs, increases the novelty. So after that, after using the model for generation, we also performed a lot of molecular dynamics simulations that I'm not going to talk about. We also performed comprehensive wet lab validation, and we set up a web service which allows the users to play with the model and use it for the design. All right, so the structure of the model looks complicated at first because we have a number of training modes and a number of objective terms, but if I decompose it now into parts, it's going to become clearer. So the model consists of, very typically for variational autoencoders, an encoder and a decoder. And the encoder takes as input a peptide, and it produces a latent representation of that peptide, which is encoded Z. And then this latent representation goes as input to the decoder. Sorry, I just want to say that this pointer doesn't really work, if there is anybody in the... Okay, so the decoder takes as input the Z, and it produces a reconstructed peptide prime. On top of this, we also have another additional latent variable, which is called C, and that variable encodes the property of that peptide. So is it active, is it inactive? So this part that I just showed you and that is highlighted in black is a very typical architecture of a conditional variational autoencoder. But when we started working with it, we realized that this standard model just completely doesn't do the job. So we really needed to extend it with additional features so that it worked for the design of peptides. And so one additional thing that we included was to repeat the encoder again, but now the encoder, which shares the weights of the first encoder, the second encoder takes as input the reconstructed peptide prime. It produces another latent representation of that reconstructed peptide, which is encoded Z prime, and we force the model to have these two latent representations similar to each other. So it stabilizes the training and really helps us to avoid collapse in the training. And following from this, we added another term to the loss function, and we called it latent reconstruction regularization. Apart from this, we did something similar in vein. So we added, to do this, we added a pre-trained model, which is called classifier. This model takes as a peptide as input, and it tells or predicts for that peptide, is it active or inactive? And the classifier is repeated again. It takes a reconstructed peptide as input, and it predicts its class C prime. And again, we have a now cross-entropy term in the loss function, which enforces that the predicted class for the input peptide and the reconstructed peptide are the same. Finally, one last extension to the model is focused on disentangling the latent representations. So the latent representation Z is supposed to encode the property of being a peptide, while the class variable is really supposed to be the variable that rules the fact, is the peptide being active or inactive? And so, does this work? Okay, excellent. Thank you. So it's now about disentangling C from Z. And the way to do this was for us to use this predicted class for the reconstructed peptide, and we computed the gradient from that class through the decoder and with respect to Z, and we set it to 0, so that the Z doesn't know about the class, it's only the class variable C. Okay? So that was about training of the model and its architecture, and now having the model trained, how do we use it in these two ways of action of unconstrained and analog generation? So for the unconstrained generation, you should focus on the yellow arrows, and this is the standard conditional variation autoencoder generation mode. So we sample from the prior. We have a slightly refined prior using PCA just to structure the latent better. But we sample a point from the prior distribution. We are getting a new point in the latent space Z, and we are sampling or putting the class by hand. And now the decoder takes these two as input, and it generates a novel peptide. So this is the unconstrained generation. But then, for the analog generation, we are putting the peptide as input. You're following the blue lines now. And so the encoder generates its representation. You're setting the class, and then the decoder generates the analog peptide. The important part here is that we added a so-called creativity parameter to the model. So the creativity parameter is modifying the variance of the latent posterior coming from the decoder. This is the distribution from which the latent representation is sampled from. And so this essentially means the larger the creativity, the further away we are searching from the original peptide representation, okay? So this larger this creativity, the more peptides can we generate, the more we can change the original peptide, so they will be more different, but then there will be more of them, they can be changed more, and they can achieve higher activity. Okay, so after I have walked you through this model, you may appreciate why the PhD student had like a resemblance of it to a monster from Greek mythology. So part of the name of the model is Hydra, and also there is a small feature in the logo. The logo also has 3 heads in it. Okay. So now, that was about the model, now about the results. So first, we tested the model in a very difficult task, okay? We gave it negative peptides as input, and we asked the model to improve them, right? To make them more active. And now, First of all, this plot is only a confirmation that indeed the peptides, the original prototypes that we took, they were indeed negative. So this is the distribution of the probability of being an AMP, so being an antimicrobial peptide, in the negative peptides test data. And this is the distribution of these scores of the probability of being AMP for those prototypes that different models were able to improve. So the red colors are for Hydrump with different creativity scores. Here we compare in blue shades to standard variational autoencoders, and then a language model called Joker. This is not a transformer model, it's just a model that traverses the space of sequences using some grammar. And now here is how this distribution has changed for the analogs. So for those peptides that we asked the model to now generate as new analogs of the prototypes, but with increased activity. And so you see that with increased creativity, Hydrump is getting better and better at producing highly likely peptides that are highly likely to be AMPs. And the median for that distribution is much higher than for the competing models. Also, another question is how many of the original prototypes is the model actually able to improve? And so, again, with increasing creativity, Hydrump was able to improve a higher and higher fraction of negatives. And so for the highest creativity, he was able to essentially improve almost all of them, while the competing methods, they were only improving up to around half of the original prototypes. So that was the prototype generation. Now about this simpler unconstrained generation. So Hydrump outcompared other generative models in this task. So we compared again to simpler conditional variational autoencoders, but also two LSTM models, another VE, and then a generative adversarial network. And the median of the probability of being highly active for the generated peptides by Hydrump was much larger than for the competing models, for which it was almost, or rather closer to zero. All right, so that was in silico validation for the, and then we moved on to actually validate our model using experiments. And so we, after training, we asked the model to produce candidates in which we synthesized in the lab, and we tested their activity and toxicity. And first of all, there are actually very few models in this domain that were published and then tested experimentally. So out of these models, we found Joker, Class, and that's it, actually. And so we also checked the AMP success rate for different activity levels, because different models consider peptides as being active using a different threshold for its minimal inhibitory concentration. So we simply checked all of these thresholds that we also used in our experiments, and we checked how many of the peptides that were originally tested had MIC lower than this threshold. And so we saw that in terms of success rate, our model outperforms other competitors for almost all of these activity thresholds, and most importantly for the thresholds that correspond to very highly peptides. So for these very, very highly active peptides, meaning very low MIC, our model was multiple times better than the competitors. So in total, so the model was managed to improve 4 active AMPs. So we, in terms of analog generation, we took 4 known AMPs and we gave it for improvement. But also, it turned one inactive peptide into multiple active ones. Altogether, we discovered 15 novel peptides, and we showed experimentally that they are not only active against model bacteria such as E. coli, but also multi-drug resistant bacteria, and we also showed that they are non-toxic. So after that exercise, we had a bunch of peptides to give them new names, and I was able to name a peptide after my cat. Okay, so that was about Hydrom, and now I want to share my perspective on this generative AI approach for discovery of new AMPs in a more sort of general view. So I really believe that generative AI is a paradigm shift in the way we are working and finding novel peptides. So please consider that the whole AMP space is really, really huge. So considering peptides up to 25 amino acids long, this space is on the order of 10 to the power 32. And then you can imagine that in this space, this huge space, there could be areas of different activity of peptides. And now the data that we actually have about them is a tiny, tiny fraction of that entire space of possibilities. And after in-depth analysis of the data that we have, we realized that it's, in fact, even could be reduced because there are subgroups of very, very similar peptides. And the reason behind this fact is the way people actually worked on discovery so far. And this is because people were taking known AMPs and they were searching in the space of sequences around them, modifying small number of amino acids and finding peptides that are very similar to the original ones. And I believe that using the AI, we are able to really move further away to another area where possibly much higher activity peptides could reside. And the way I tend to think about this problem is using an analog to like an entire galaxy where you would have these different AMPs as different stars in the different areas as different solar systems. And so I believe that using generative AI, we could have like this spaceship that would take us to another star system to better AMPs. And so the way ahead for me is now to continue working on this project in a novel project actually called DOGAMP, where we are working on deep optimized generation for antimicrobial peptide discovery. So here we are really focusing on the word optimize. Okay, so we instead of only teaching the models to generate, we also want to teach them to optimize the peptides. Okay, we want to optimize them in terms of their activity, and we want to optimize them in terms of
toxicity and other features. One small step in that direction is our recent work called Joint Former, where we are teaching the model both generation and prediction capabilities. So we are merging BERT and GPT, if you like. So we are switching, doing training, the multi-direction and causal attention, and so that the model actually learns the joint distribution of the property and the peptides themselves. And so I believe that using generative AI, we can really make a step forward in addressing antimicrobial resistance. Hydrom already showed us some promising results. We are heading now to optimize generation tasks. And so I hope for this spaceship to another star system in the peptide galaxy. And that's it about the antimicrobial peptides. I just want to give you two announcements. One is that I'm organizing or co-organizing a foundation model in health symposium in Munich. So we have a number of wonderful speakers. If you're interested in that topic, please, there's still possibility to register, and you can come and visit us in Munich, April 10th. Another one is that I'm actively looking for PhDs and postdocs. If anyone is please reach out to me or just apply. I just want to acknowledge people in the lab that did the work. Okay, so Marcin Morzejko and Paulina Szymczak are the first co-authors on the Hydrom paper. I want to thank my amazing collaborators and my funding, and that's actually now is the finish. And thank you for your attention.